This video will be all about how bacteria interact and some of the interactions that occur among bacteria. And then we'll look at sporulation, the process of making an endospore. So, if bacteria are found in sufficient numbers, then something really cool can happen. These new metabolic functions in the bacteria can be initiated, and that can lead to the production of bioluminescence, which is when the bacteria produce their own light as they perform certain chemical reactions that produce that light. We can see the production of toxins, of digestive enzymes, and of adhesive molecules, molecules that make them really sticky. So like just a real sticky substance. And they know each other. They know that there are sufficient numbers of bacteria to bring on these processes through a process called quorum sensing. So a quorum in, if you think about like if a group of people are voting on something, there frequently has to be a quorum present in order for there to be enough people to vote. So that's the idea of quorum sensing, is that the bacteria know when there are enough individuals around to make doing these processes here worth doing, because there'll be enough bacteria that they can actually be effective in producing bioluminescence or producing toxins. So quorum sensing is effected through the production of molecules that are referred to as inducer molecules. And as there are enough bacteria to produce a sufficient level of inducer molecules, then that is what allows the bacteria to sense that a quorum has been established, that there's enough bacteria. And once there is a sufficient concentration of these inducer molecules, then we see the new metabolic function, whatever it is, bioluminescence or making these sticky adhesive molecules, that triggers the genes that let that behavior, that new behavior, turn on. Okay, so one example of this kind of quorum sensing that is posing a big problem in hospital settings and other healthcare type settings is the production of a biofilm. So biofilms are produced in response to quorum sensing. And here is a close-up view of what a biofilm can look like. This is Staphylococcus aureus, the same species that we have strains of that are methicillin resistant, and so we call it MRSA. That's what MRSA stands for, is methicillin resistant Staphylococcus aureus. And so here is a biofilm by scanning electron micrograph of a biofilm, and you can see all the little Staph aureus cells, and then you can see that gooey substance that they're all trapped in. That's the biofilm that includes the bacteria. So that's that gooey, sticky stuff that they make. So here's how biofilms are established through quorum sensing. So as individual cells settle on and adhere to a surface, they start producing that inducer molecule. The cells grow and divide, and as more bacteria grow, then the inducer molecule begins to be produced in greater and greater amounts. And that inducer molecule is what then establishes that the biofilm genes get activated. So in response to these genes, or as the genes are activated, then the bacteria begin to produce this really slimy matrix of proteins and DNA and sugar, and they become embedded, so they secrete this stuff all around them, and they become embedded in this thin film of extracellular gooiness. And the bacteria that are found then in the biofilm, they develop different capacities or different abilities, as well as producing that biofilm stuff. Those nearest the surface nearest the surface that they're growing on. So the ones that are deep underneath this gooey biofilm, frequently those will develop the ability to resist many antibiotic treatments. And then other bacteria will develop other functions. The book talks about how some become really good at utilizing oxygen, and so others will develop other functions, but frequently it's the ones that are nearest the surface that was colonized that become very resistant to any antibiotic materials. And because they're embedded in that extracellular gooiness, they can also be very resistant because of the protectiveness of the goo. They can become very resistant to any antiseptics that you spray onto the surface. So where do biofilms develop? Well, they can form on wounds, and if you can take it on figure 10.9, so you have to go forward to chapter 10, but figure 10.9 shows a wound that had a biofilm associated with it. It's in a person with diabetes who already has poor circulation, so it's a wound on the leg. The person had this wound for 10 years, trying to treat it with all kinds of antibiotics, no effect, and finally they used a phage, a bacteriophage, to treat it, and they finally were able to heal the wound. So biofilms can form in wounds. They can cover medical equipment. They are what becomes plaque on the teeth. So as somebody doesn't brush their teeth, then a biofilm can develop, and then that biofilm can even be calcified over time because of the calcium salts that are in saliva, and so that becomes the plaque that then the dental hygienist has to chip off your teeth. And any wet surfaces, like in the pet's water bowl. I frequently find biofilm in my cat's bowl. That's how I know it's time to wash the cat water bowl. And because these biofilms can have in them not genetically resistant to bacteria, but biofilm-related resistance to bacteria, biofilms can pose a significant risk to humans, especially in hospital settings. And that's why we take extra care that anytime, like an implant, like a knee replacement, where they're putting equipment inside the body, they take super extra care to make sure that they are sterile pieces that are going into the body. Okay, so biofilms are one way that bacteria interact with each other. The book has some other examples, including like zones of inhibition between colonies on agar plates and some other ideas. So I want to encourage you to read the book, as always. So let's talk a little bit about sporulation now, too. Endospores, the internal structures that form inside a bacterium, usually, as conditions in their medium become less and less favorable, endospores are formed through a process that's named sporulation. And we see endospore formation in gram-negative bacteria like Bacillus and Clostridium, as well as gram-positive bacteria. Several gram-positive genuses or genera. That's the plural of kingdom, phylum, class, order, family, genus. That's what that Bacillus and Clostridium, those terms are the genus names of bacteria. So unlike spores that are seen in kingdom fungi and kingdom plantae, bacterial endospores are not about reproduction. So in most bacteria, they are not about reproduction because one bacterium can only produce one endospore. And with reproduction, you would expect that there would be multiple spores being produced by one bacterium. So rather than being a reproductive strategy, sporulation is about surviving poor conditions. So as resources are being used up in their environments, sporulation is triggered, and the bacteria develops now this this protected copy of its DNA that will last through the bad times. So the bacterium will die, but it will live on through the endospore. And as we've talked about, endospores can survive for centuries and are super resistant to extreme temperature. They're resistant to pH, to the effects of radioactive materials. So they're resistant to radiation. And they're even resistant to some toxic chemicals. So super resistant to all kinds of stresses that will allow the bacteria then to come back to life later on. So the process of sporulation takes about 7 hours. So as conditions are getting icky in their culture, then 7 hours later you can start seeing spores appearing in the bacteria. So the first thing that happens in the process is that the DNA is replicated, and rather than forming the usual kind of loose chromosome in a nucleoid, this replicated DNA is a long and very compacted form of DNA, and it's referred to as the axial nucleoid. So we produce this secondary bit of DNA called the axial nucleoid, and then from there on, the axial nucleoid, that DNA, is what directs the processes of spore formation. So a lot of the RNA in the cell and some of the proteins that are found in the cytoplasm then kind of cluster around that axial nucleoid, and that forms what is referred to as the core of the endospore. So here the axial nucleoid is forming along, so here's our usual nucleus over here if we look at it. And the let's see. Yeah, where am I? Okay, so the axial nucleoid forms and is in its own little separate region of the cell. And so as the RNA in the cell, the proteins in the cell all cluster around it, that forms this endospore core. And in the core is a special organic molecule called dipleyonic acid and calcium ions. And those two materials stabilize the protein, and they are responsible for making it resistant to heat. So dipleyonic acid and calcium are what provide the heat resistance that we see in the spore. Okay, so we'll continue on. Next, a septum begins to form. The septum is a double layer of plasma membrane, and in between the two layers of the plasma membrane, we see a lot of peptidoglycan being formed. And so it fills up that space between the membranes. And so the membrane then, in that peptidoglycan layer, becomes what is referred to as the spore cortex. So you have a core, which has got DNA, RNA, proteins, dipleyonic acid, and calcium in it, and then a spore coat the cortex of the spore, which is this double layer of membrane stuffed with peptidoglycan. So now core, cortex, then around the cortex we see the development of a spore coat. Now the mother cell, rather than the bacterial DNA, is what directs the production of the spore coat. And this coat is this keratin-like protein that wraps around, so we get multiple layers of this keratin-like protein, and it forms then the exosporium, which is this. I'm sorry, I take all that back. Let me try that again. So the mother cell produces a spore coat of keratin-like protein, and then there's even another layer on top of that, the exosporium, that's got a lot of lipid protein material. So it's another membrane of lipid and protein around it. So now if we're looking from the inside out, there's the core, the peptidoglycan and membrane cortex. There's a spore coat, which has this waterproof keratin-like protein around it, and then external to that is the exosporium with that lipid protein membrane. And so once sporulation is complete, then the mother cell lyses and dies, and now the spore is released to the environment, and it is going to be able to resist drying and heat and acid and base and radiation until favorable conditions return. So when favorable conditions finally return, spore germination, or the conversion of this dormant spore to a growing vegetative cell, occurs in 3 stages. There's activation, and this usually requires some sort of traumatic event that damages the spore coat. So activation might happen as pH changes in a significant way, or heat happens, or something that damages the spore coat so that the delicious nutrient-rich fluids can get into it. So as water and some germination agent, and it just depends on what it is, alanine, which is an amino acid, inorganic ions, so stuff in the medium around the bacteria spore, as those things begin to penetrate into and through that damaged spore coat, then the peptidoglycan and the cortex break down, and then water can flood into the core, and the spore now begins to metabolize. And at this point, the spore is no longer resistant. So if something damages the cell now, the spore at this point, once germination proper has begun, then the spore has lost its resistance. So then last of all, we see outgrowth. This is when the spore, or the now growing vegetative cell, really begins to become metabolically active as RNA is being produced, and then proteins are being built from the RNA. And so within about an hour after we see RNA being produced, then DNA synthesis begins. And so it starts to make a new chromosome, and binary fission will proceed. So sporulation, 3 steps of activation, germination proper, and then finally binary fission that tells us that the colony of bacteria will be growing again now. Okay, so there you have it. All the processes of sporulation, plus the idea of quorum sensing and the ability of quorum sensing to lead to the production of these biofilms that can have hazardous consequences in healthcare settings. You know what to do.